Good morning, all of you. In the last class, we have been discussed regarding systematic classification types, natural classification, artificial classification, phylogenetic classification. We also discussed, and as well as organisms classification also we have been discussed. The first one is two kingdom classification proposed by Leibniz, and three kingdom classification is Haeckel. Four kingdom classification is proposed by Copeland. Five kingdom classification is proposed by Whittaker. So till five kingdom classification we has been discussed. Today I am going to start sixth kingdom classification. Sixth kingdom classification. Sixth kingdom classification. Here, the sixth kingdom classification is proposed by Carl Woolf. Proposed by Carl Woolf. So, according to the Carl Woolf, all organisms are grouped into six categories. The first category is Archaebacteria. Second one is Eubacteria. And third one is Protista. And fourth one is Fungi. And fifth one is Plantae. And sixth one is Animalia. The sixth kingdom classification is proposed by Carl Woolf. His categories are all living organisms into six kingdoms or six categories. The first one is Archaebacteria, Eubacteria, Protista, Fungi, Plantae, as well as Animalia. So here the question is comes here: why Carl Woolf separated the Archaebacteria from Eubacteria? What is the main reason? And what is the difference between Archaebacteria and Eubacteria? So let's see the difference between Archaebacteria and Eubacteria. Archae means older. So this category is the oldest bacteria. The oldest bacteria are grouped in Archaebacteria. And here U means true bacteria. It means true bacteria. And here Archaebacteria are here, these bacteria also contain cell wall, but this Archaebacteria cell wall is not made up of polysaccharides, not made up of polysaccharides. The cell wall is made up of pseudo-murin. In Eubacteria, the bacteria cell wall is made up of polysaccharides, and murin or muric acid. Right. And one more difference is there in between Archaebacteria and Eubacteria, that is here RNA polymerase. Such compounds, RNA polymerase compounds or subunits are complexly arranged. The subunit arrangement is very complex. And here RNA polymerase subunits are Simple arrangement. RNA polymerase units are RNA polymerase units are simple arrangements we can see. So this is a major difference between archaebacteria to eubacteria. So that's why God knows he separated the oldest bacteria from eubacteria or nothing but true bacteria. Example, Methanomonas bacteria. What is Methanomonas bacteria? What is the functions? We will discuss when we enter into the Munira. Next, eubacteria. Eubacteria is not all bacteria like Bacillus, right? Bacillus. Okay. So that's why he separated the archaebacteria from true bacteria, nothing but eubacteria. And actually, he says that six kingdom classification, it is actually cluster into three categories. So these three categories are, the first category is, as well as you know that archaea. Categories is nothing but domain. The first domain is archaea. Second domain is eubacteria, nothing but bacteria also you can write. And third one here we have to focus on eukarya. And in archaebacteria, he is included. Oldest bacteria is included. In eubacteria, he is included bacteria and as well as BGA, blue green algaes. The blue green algaes which have the capacity to perform photosynthesis. What are the examples of BGA? We will discuss when we enter into the Monera kingdom. Next, eukarya. Here in these all eukaryotic organisms are included. All eukaryotes are like plants as well as animals is included. Now it has been discussed regarding 2 kingdom, 3 kingdom, 4 kingdom, 5 kingdom as well as 6th kingdom classification has been discussed. But in the NCERT, they mainly focus on 2 kingdom classification as well as 5 kingdom classification. And the chances is there to ask the 2 mark questions as well as 3 mark question from 2 kingdom classification as well as 3 kingdom classification. Let's see, one question is, what are the demerits? What are the demerits of Linnaeus classification? What are the demerits of Linnaeus classification? See here, what is the major demerits of Linnaeus classification? The first part is the classification is a artificial classification. It is an artificial classification. He is considered only 1 or 2 morphological characters is considered. Only 1 or 2 morphological characters is considered. And according to the NCERT, the third part is given in the NCERT is not distinguishable. He is not distinguished between eukaryotic to prokaryotic, and unicellular and multicellular and photosynthetic to synthetic or photosynthesis to non-photosynthesis organisms. He is not give the difference between unicellular and multicellular photosynthetic organism to non-photosynthetic organisms and eukaryotic to prokaryotic. Clear? So this is the major three demerits of lineage classification. Next, what are the merits of Whittaker classification? What are the merits of Whittaker classification? And you know that we have discussed Whittaker classification. The first is considered maximum characters is considered, and second, not only morphological character, he is also considered chemical combination of cell wall. Chemical combination of cell wall also is observed. Next, and also is considered cell organelles like nucleus, prokaryotic nucleus as prokaryotic cells as well as eukaryotic cells. Like, and as well as mainly this important point is he is considered the systematic. He is in the classification is for the evolution pattern he is followed. Nothing but phylogenetic relationship between one species to another species. Clear? So that's why Whittaker classification is the most acceptable classification. It is considered maximum main characters is considered. So that's why almost all living organisms are covered in this classification. Clear? So this is merits of Whittaker classification, demerits of lineage classification. Next here Whittaker classification. In NCERT, detailed information is given regarding Whittaker classification. Whittaker classification. When Whittaker proposed a classification, the criteria of classification is changed. Say for example, earlier, before the Whittaker classification, how the scientists were classified the organisms. Earlier, the scientists are grouped. Or brought together bacteria, BGA, growing organs, algae, and angiosperms, and gymnosperms, and gymnosperms. So earlier, the scientists brought together bacteria, BGA, algae, and angiosperms and gymnosperms. These all are they considered as one group. These all belong to one unit, one group they consider. Clear? And also unicellular as well as multicellular organisms are the grouped into one category or one group. For example, unicellular organisms. Here, Chlamydomonas as well as multicellular, Spirochaeta. So no need to worry about this. When the new words are came in the lesson, no need to worry. This we are going to discuss further. Say for example, Chlamydomonas, it is an algae, unicellular flagellated motile algae. Spirogyra, it is also algae but multicellular non-motile algae. So simple diagram. So this is chlorella. So these are flagella and unicellular. Spirogyra. Here it is multicellular. So earlier they grouped unicellular organisms as well as multicellular organisms, they grouped into one category. Clear? And as well as prokaryotic cells and eukaryotic cells. Prokaryotic cells as well as eukaryotic cells, they are also grouped into one unit or one category. And also here Chlamydomonas, and chlorella. These two are Chlamydomonas is algae, chlorella also is algae, unicellular. So these two are they grouped into plantae. They considered as plant. And next, amoeba as well as paramecium, they considered as animals. So this is a classification. This is an earlier classification before the Whittaker. But when Whittaker is entered, the criteria of classification is changed. He cleared the issues in classification and problems. See here, Whittaker first was separated the prokaryotic organisms from eukaryotic. Organisms. And also, he separated the prokaryotic cell grouped into monera. Next, and he also separated a unicellular organisms from multicellular organisms. See, before that, unicellular organisms as well as multicellular organisms, they both are grouped into one category. They are considered as one unit, one category. But here, he separated the unicellular organisms from multicellular organisms. So, these unicellular as well as eukaryotic organisms are grouped in Protista kingdom. And as well as he cleared one more doubt also here. Here Chlamydomonas as well as chlorella, before they considered as plants and they grouped into plantae or plantae kingdom. Amoeba as well as paramecium both are considered as animals. They grouped in Animalia. But here what he did, Whittaker. Here he observed Chlamydomonas, chlorella, amoeba, paramecium. These all are unicellular as well as eukaryotic cells. These unicellular as well as eukaryotic cells are grouped into Protista. These all are coming under chlorella, Chlamydomonas, amoeba, and paramecium. So he says that these all are grouped into only one category, that is Protista. And you know very well what is the main character of Protista? Which organisms are unicellular as well as eukaryotic. Such definitely belong to what? Protista. Chlorella also unicellular, Chlamydomonas also unicellular, amoeba, paramecium, all are unicellular and eukaryotic. So that's why these all are grouped into Protista. So this is actually how Whittaker is cleared the issues and problems in classification. Clear? NCERT table number 2.1, we take a classification. We take a classification. So here, the Whittaker classified the all living organisms into 5 types based on these major 5 characters. The first one is cell type, cell wall present as well as absent, and cell wall chemical combination also is considered. Next, nucleus membrane absent or present, and body organization nothing but thallus organization. Next, mutation. Based on these 5 characters, Whittaker classified the all living organisms into 5 kingdoms. Here, Monera, Protista, Fungi, Plantae, as well as Animalia. Then you can write them down also. We can write the Animalia or Plantae here, or we can give the Plantae. We can give the position of Plantae. We can change or not. Like question we can raise in the mind. Here, this is a order is a phylogenetical order. Here, Monera is kept in first position because Monera kingdom have a primary or very primitive characters are there. So that's why the Whittaker is given the position to the Monera is first. And Animalia is very complex organism and advanced characters are there, so that's why in the last position of Animalia. So it means this arrangement is what? This is a phylogenetic relationship or a phylogenetical relationship. See, Monera cell type. First we observed cell type. Cells are mainly 2 types based on the nucleus. Eukaryotic cell as well as prokaryotic cell. Monera is a prokaryotic cell, and Protista is a eukaryotic cell. Fungi also eukaryotic cell, and plants also eukaryotic cell, and animals also eukaryotic cells. And you know the difference between prokaryotic cells as well as eukaryotic cells. Prokaryotic cell means the nucleus should not covered by any nuclear membrane. Nucleus envelope is absent. So such are called as what? Prokaryotic cells or prokaryotic nucleus. In eukaryotic characters what? The nucleus is covered by one nucleus envelope or nucleus membrane. So these are what? Eukaryotic cells. Next, cell wall. First we have to see cell wall is present or absent. Cell wall in Monera, cell wall is present, and here Protista also cell wall is present. Most of the cases present. And we said in Protista, cell wall character is not a defined character. Cell wall may be present, also absent. Not a defined character. Present may be present or absent also. Maybe absent also. Fungi, cell wall is present. Plantae, cell wall is present. And animal, cell wall is absent. Cell wall is absent. Next we have to see cell wall chemical combination we have to observe. Monera. In Monera, cell wall is made up of polysaccharides, especially we can write bacteria cell wall is made up of peptidoglycan or uric acid also we can write. And inside is given beside the polysaccharides, amino acids also present, acids also present. Next, Protista. So it is not a different character. Next, fungi. Fungi means cell wall is made of chitin. The fungi cell wall is made of chitin. And plantae cell wall is made of cellulose, hemicellulose, pectin as well as lignin. Pectin and lignin. So the lignin as well as pectin, these combinations we can notice in the toughest plant in gymnosperms we can notice. Next, animals and bodies absent. Clear? Want to discuss? Next, nucleus membrane. Nucleus membrane here, nuclear membrane here, Monera is absent because it is a prokaryotic. Here, Protista is a eukaryotic, nucleus membrane is present. And here in fungi also eukaryotic, it means nucleus membrane is present. And plantae, here also present. And animals also eukaryotic, nucleus membrane is present. Here, next, body organization, nothing but thallus organization. Thallus organization. One more question. What is meant by thallus? Thallus is a undifferentiated, it is an undifferentiated plant body, or we can write it is not differentiated into as a true stem, leaf or root. Such an undifferentiated organism is called as what? Thallus. Thallus are like thallophyta. Which organisms are containing thallus? Such as belong to thallophyta. Algae belong to thallophyta. Fungi belong to thallophyta. Next, Monera body organization is. Monera is a bacteria, single cell, it means cellular, or you can write single cell. And the body organization of Protista. So we already given information regarding Protista. Protista is eukaryotic as well as unicellular. Eukaryotic as well as unicellular. In fungi, some of the fungi, only few fungi are unicellular, what we can call as cellular body, and some fungi are tissue. Tissues are present. The body organization is now developed from its advanced character. For example, see at the beginning, only one single cell primitive character, and you can see the second thal. Here we can see tissues, little bit advanced character. So this arrangement is called as phylogenetical arrangement. So tissue loose tissues we can notice in fungi. Next, plants. So plants are multicellular. Here we can notice cell to tissues, tissue to body, and body parts also be found here. And animalia are animals here very advanced here. Cell to form, cell to cell, tissue to organ, organ to organ system. Here organ system also we can notice. So whatever he is in the position according to the character, primitive character to advanced character. Next, nutrition types. Nutrition type is here. The first one is Monera. Here, Monera, it can show the maximum types of nutrition it can show. The first one is generally most of the bacteria or Moneras are heterotrophic. And some are parasites, and some are saprophytes, and some bacteria also have the capacity to perform photosynthetic. We can call as chemosynthetic bacteria. Photosynthetic bacteria also we can call. So here, what is chemosynthetic? So these bacteria These bacteria can prepare the organic substance by using of energy which is released during the chemical reaction. During the chemical reaction, some energy is released. So such energy is utilized for synthesis of organic compounds. So such are chemoautotropes we can call as. Chemoautotropes we can call. Clear? What are the chemoautotropes or autotropes? We will discuss when we enter into the Munira kingdom. So this is a maximum types of nutrition it comes to the Munira. Next, protista. So protista here it is also the nutrition. It is a non-defined character of protista. Here some are autotropes and some are heterotropes. So also the galactic protista. Earlier we have to discuss Chlamydomonas, chlorella, both are what? Photosynthetic organisms. Paramecium as well as amoeba, both are non-photosynthetic. But these paramecium, amoeba, chlorella, Chlamydomonas, these all are grouped into a protista. Clear? Next, fungi. Fungi it is a non-photosynthetic. It is mostly saprophyte. Saprophytic nutrition. Plant, you know that these are photosynthetic. Photosynthetic organisms. Autotrophs, mostly autotrophs. And here animals are heterotrophic halozoic organisms. Clear? So this is the information is given by Whittaker classification. So let's see how this classification is going to be useful for classification of all living organisms. For example, here I am going to give one or two example how this classification is practically worked out to classification of organism. For example, one scientist is noticed one discovered one organism that is containing cell wall. It is containing cell wall, but the nucleus is do not cover nucleus membrane. Nucleus is do not cover the nucleus membrane. So such organisms where we are going to group according to the Whittaker classification, cell wall is present, but nucleus membrane is absent. So it means based on these two characters, according to the Whittaker, we are going to group this in Munira division. For example, some of organisms cell wall is present. Cell wall chemical combination is cellulose, and our nucleus type is eukaryotic. Then these organisms were grouped into plants. For example, one more example, cell wall is present, but in the place of cellulose, chitin is present, and nucleus is a eukaryotic nucleus. Then where we are going to group it? We are going to group it into fungi. So based on these characters, also we have to consider like nutrition also we consider, because the fungi the main function is the main character is saprophytic nutrition. Clear? So in that way, the Whittaker is going to clear the issues as well as problems during the classification of organisms. So this is complete entire regarding Whittaker classification. One maybe the chances is to ask 5 more questions also. In the next video, we are going to discuss regarding Munira. Thank you.